Hello friends, welcome to BMH Learning. In this video, we are going to talk about microalgae isolation techniques. There are different techniques for the isolation of microalgae. Let's start with serial dilution. Equipment and materials required: sterile culture tubes, test tube racks, nutrient media, 10 ml pipette, 1 ml pipette, Bunsen burner. Procedure: take 5 test tubes. By following aseptic technique, add 9 ml of media into each of 5 test tubes using sterile 10 ml pipette. Label tubes 10 raised to power minus 1 to 10 raised to power minus 5, indicating dilution factor. Then add 1 ml of enrichment sample to the first tube and mix gently. Now from the first test tube, take 1 ml and add to the next tube and mix gently. This makes it 10 raised to power minus 2. Repeat this procedure for the remaining test tubes, that is up to 5th tube. So the 5th tube is most diluted. Incubate test tubes under controlled temperature and light conditions. After 2 to 4 weeks, we can examine the microalgal cultures using a microscope by aseptically withdrawing a small sample from each dilution tube. You may observe a unialgal culture growth in one of the higher dilution tubes. In case of the tubes containing different species, that is more than one, then you can use micromanipulation to obtain unialgal cultures. Next method is streak plating. This method is best suited for small species or for those species that grow well on a substrate. Equipment and materials required: sterile petri dish, nutrient media, wire loops, Bunsen burner, parafilm. Procedure: petri dishes are prepared containing growth medium that is solidified using 1 to 1.5% agar. Note one thing, that agar should be 1/2 to 2/3 the depth of the dish. Now 1 to 2 drops of mixed phytoplankton sample are placed near the periphery of the agar. Using a sterilized wire loop, aseptically make parallel streaks of the suspension on the agar. You can notice there are 16 streaks, so that means we have made 4 sets of 4. This way, the whole surface of the agar plate is used. The petri plate is then covered and sealed with parafilm. Invert it and then incubate under low light at constant temperature. After incubation period is over, you can observe the algal growth. We have to select those colonies which are free of other organisms for further isolation. So a sample is removed using a sterilized wire loop and placed in a drop of sterile culture medium on a glass slide. Then we check with the microscope if the species are actually isolated and if they are unialgal. Now repeat the streaking procedure with these isolated unialgal cells coming from a single colony, and the colonies are allowed to develop again. Now why we are doing second streaking? It's because the second streaking actually reduces the chances of bacterial contamination and also minimizes the possibility of colonies containing more than one algal species. This was all. Thanks for watching.